I'm from Paris. I'm working at Universite Paris Descartes at the Integrative Neuroscience and Cognition Center. So this talk will be about mainly neuroscience. And so what I will, in a nutshell, what I would do is I will show a way to find a neural correlate of consciousness awareness on the base of neural synchronization. So I will have to explain what is neural synchronization. And the basic idea is that we use a large data set of epileptic seizures in epileptic patients, and also people during sleep, during different phases of sleep. That was a data set obtained during several years, and it was not done for this work specifically, but we analyzed it a posteriori. And we obtain a correlate of conscious awareness that is based on the construction of a functional network. So a functional network is the network of the connectivity, of the functional connectivity between brain areas. And we have, it's very simple, actually it's a very simple way of computing. We did a very simple data analysis on this data, and is simply to calculate the information content of this functional network. And this is what I'm going to explain. But before, I want to talk about neural synchronization. So I like to think about this in terms of segregation and integration in the brain. And a nice analogy is this of an organism, a crab here, that is trying to move, and you see the locomotion, and perhaps is also eating, and there are different parts. So there is specialization, but they have to be coordinated somehow. So that will be some sort of temporal coordination that is important for the organism. And somehow something like that is happening in the brain also, because there is segregation. So that would be analogous to the specialization. And information is processed in different parts of the brain, but it has to be integrated somehow. And we claim that, not me, but it's a hot topic in neuroscience, that this integration is happening through neural synchronization. So I would first say that synchronization is happening at multiple scales. So you can have synchronization between a pair of neurons, but you can also have synchronization in cortical columns, or even long-range synchronization between different brain areas. So it's a widespread phenomenon in the brain, and it is supposed to be a basic mechanism for integration. Actually, some people go a bit beyond that, and there is this binding by neural synchrony hypothesis that maybe they claim is the solution for the binding problem, although not everybody agree on that. So another point I want to make is that synchronization is a universal phenomenon. So it's not limited to the brain, it's not limited to neurons. It's really something that happens whenever you have nonlinear couple oscillators, weakly couple oscillators. So this is a study in nonlinear dynamics. It's a very huge field of nonlinear dynamics and physics, and therefore you have examples in chemistry and biology. For example, this famous fireflies and so on, like the Millennium Bridge, for example. You have lots of examples, and I want to make one simple example. Is this one? So you take a table on cans. The cans are allowed to move, and on top you put two metronome clocks, so that's pendulum clocks, and then you put them in different positions and wait some time, and eventually they are in phase or in antiphase, so they are synchronized. So it's a very, I want to emphasize this is a very basic physical phenomenon, and it turns out that if you want, you can think about neurons as oscillators in certain instances, and that this has been found, for example, because neurons spikes, and the spikes can be model like dripping faucets, which is a self-sustained oscillator. And when you have self-sustained oscillators that are coupled, or weakly chaotic oscillators that are coupled, they typically synchronize. So this is a very well understood phenomenon. And not only neurons can synchronize, but also populations of neurons can synchronize. And in some instances, you have actually macroscopic oscillators, essentially. So this can happen during epilepsy. And this is what I'm going to show you. Actually, even if I said that most probably synchronization, neural synchronization is a mechanism for integration, too much synchronization is bad, actually. And there are several cases that you can study about this. And one specific, very clear, is epilepsy, but also Parkinson's disease and others, in which too much neural synchronization leads to a pathology. In particular, if you see a patient during a partial seizure is a seizure that is focal, so it's located in some place in the brain. If you look at that, and you look at the electric activity, is measured with electroencephalography, for example, you see that in different channels, so the channels are electrodes, you have, before the seizure, you have the standard activity that is very noisy, but during the seizure, you have a very rhythmic oscillation. And this very rhythmic oscillation is also synchronized between different areas. So you have the same, essentially the same activity in different parts of the brain. In the case of a generalized seizure, so the seizure has gone in the whole brain, you have the same activity in all the channels of the brain. And interestingly, this leads to loss of consciousness. So which is why we were interested in that in terms of consciousness. And when I say consciousness here, I really mean consciousness in a very medical, almost sense of the word. So like, this is loss of consciousness. Yeah, really, they can be on the floor like this. So a question that arises was, is it that synchronization is directly related to consciousness? And this is a question that goes beyond, perhaps beyond consciousness, but it's important in a medical setting, because it was observed that for many diseases, what you observe is the lack of variability. For example, you can see that in heart rate variability, and if it is lack of heart rate variability, then you have some pathology. And this is also the same in epilepsy, because the whole brain essentially is synchronized, and that's why you have tremor during epileptic seizures. And it turned out that the answer is not that simple. It's not that more synchronization is bad, because we also know that there is necessity of synchronization for integration. So this was some kind of paradox. So what we did was construct the complexity measure. So there was something missing here, and we thought it was complexity. And the complexity we built was strictly based on synchronization. This maybe is a bit different to other complexity measures that had been obtained. And it's not based on machine learning or any kind of like black box approach, but it's based on physical principles. And well, we work with this kind of devices like MEG and EEG, or intracranial EEG also. So like in this case, you have electrodes inside the brain. And what you want to calculate is essentially how much these signals are oscillating in phase. So like if they were sinusoidals, it would be a very easy task. You will see that they are synchronized. But these are typically noisy signals, so the point is how to obtain this amount of phase synchronization, phase coherence, or it's a statistical measure. So like here is more schematically, you have channels outside of the brain. This is scalp EEG, for example, or MEG. They are measuring electric or magnetic activity from inside of the brain. So you have two signals, X and Y. And the point is how to measure the phase of the signals. This is a very, very, very standard problem in signal analysis, so I will not discuss it in detail. So one important thing is that you have to filter the signals to make a sense of it. And then you construct essentially something called analytic signal to obtain the phase, which I can very easily give you an intuitive idea. It's like you have two real signals, so they are real numbers in time. And what you do is convert them into complex numbers. So like then eventually, immediately you have a phase. So what you see is like if they're same angular velocity instantaneously, then they are synchronized. And then you do that for like chunks of data, and simply you compare that. It's very easy to do. And so like it's here. You obtain this index. This is the phase difference between the two signals. And this index is called mean phase coherence. And it's a number that goes between 0 and 1. So 0 if they are not synchronized, and 1 is they are completely synchronized. So it's very, very easy to do. It's standard techniques day by day. We use it all the time. And one thing we wanted to do is like from there obtain a complexity measure. So like we wanted to have the ability to say two electrodes are coupled or not. So we converted these values into Boolean values, so 0 or 1. 1 if they are connected, 0 if they are not connected. Very easily done by surrogate analysis, which is essentially take the time series and randomize them. So you randomize the time series, keeping the spectrum, but randomizing the phase in such a way that you break all the correlations. And in that case, you obtain a value of synchronization, small r, here, small r. So this is called threshold analysis. You have a threshold. This is what you take now as a basis to say this is synchronized or not. So if something is below that threshold, you put a 0. If it's above, you put 1. And then you have, between these two channels, if they were sinusoidals, we'll have 1. If it's really something very noisy, then you have 0. And you can do this channel by channel, so couples of channels by couples of channels all around the brain. And you have matrices, like these are channels here, and you have 140 channels against another 140 channels, like you compare, you do pairwise correlations. And what you get is like a connectivity network or connectivity matrix, as you like to call it. It is full of zeros and ones. These are examples from real data. So this is before the seizure. You get something like this. And during the seizure, you get something like that. So you see before seizure is full of zeros because there's not enough synchronization. So things are not coupled. And then during the seizure, this is some sort of storm of synchronization. You have this matrix full of ones. And so on. You can have these at different times. But I want to compare these two specifically. And now what you want to do is a measure of complexity. Essentially is to measure the complexity of these matrices. So I can convert them like into a string of zeros and ones. And simply count the possible number of configurations of these matrices given a number of ones that is telling you how much the system is globally synchronized. So how many couple of channels are coupled is telling you a global amount of synchronization. So given this amount of synchronization, how many patterns you can have. And of course, if you have a state where everything is synchronized, like here, the matrix is made of ones, you have only one state. If you have a completely non-synchronized system, again, you have one state. And only when you are somewhere in the middle with some ones in the matrix, so intermediate levels of synchronization, then you have many patterns because you can simply switch, shift the ones in different positions. And then you count the number of patterns, and if you do this calculation analytically, simply like it's some combinatorial problem, you have some inverted U curve telling you the number of possible configurations as a function of the number of connected pairs if you want global synchronization. So you see that as synchronization is increasing, it goes through a maximum. So like at some intermediate value, there are more states. So now if you do a small assumption, well, not actually small, it's a bit brute assumption, which is that pairwise configurations are equiprobable. It's a bit like not necessarily true. We didn't do the experiment to know which are the probability distributions of this configuration. So we cannot do it actually. We cannot ask a person to have the same seizure 1,000 times. So that's not really possible. So we do some kind of trick, which is we assume that they're equiprobable, and then what we are doing is counting states. If you take logarithm, this is the Shannon entropy. So in that case, we can give, let's say, some interpretation to what we are doing, which is that we are calculating the information of the network. That's important. It's not the information of the signals themselves, but of the network of connections. If you do it with the signals, you don't get nothing. You don't get anything interesting. So I give you an example. This is a seizure. During a seizure, before the seizure here, you have the typical activity of the brain. It's almost noise, pure noise, 1/f noise actually. And during a seizure, you have an increase in amplitude and very rhythmic activity. And if you look at entropy, you see like entropy is decreasing all of a sudden. So there is a decrease and then an increase in entropy. And if you also now do the same, but like take pieces, like I take a piece in what I call the baseline, which is where the person is in a normal stage. And then I take the piece of the seizure and I want to compare the two values of synchronization. Then you get something like this. Sorry, this is a very, very difficult to interpret graph. So there are four different patients. So these are different patients. The techniques used were different also. One was intracranial EEG, the other was EEG, so on. So they are very different. The data is very heterogeneous. So like it's not always the same technique, always the same person. It's like we have a very heterogeneous data set. But what we see, the main idea is that during baseline, entropy is higher than during seizures. And there are two exceptions here. Here, the baseline is below the seizure, and the same here. But it turns out that these are patients that were conscious. So they were alert, they were responsive. This is a 10-second seizure, and the patient was responding. And here is a non-generalized seizure. Again, it was not unconscious. How much time do I have? 1 minute 25? Okay. So the same, I will not go in detail, but it's essentially the same in sleep data. Sorry, there's a very confusing graphs, but look at awake here. So this is data when the patient is awake. Compared to REM, rapid eye movements, you have dreams, it's a bit lower. And compared to slow wave in sleep, you see slow wave 2, slow wave 3, 4. These are, you are going down in the amount of consciousness. And again, you are going down also in entropy, right? So the whole thing makes sense. And to conclude, this is the picture I would like to show. It's like conscious or alert states are more like the tip of the curve, like they correspond to intermediate values of synchronization. No low, no high. So high and low stages are low entropy. It's a global measure, is in the network. We can't say anything about it locally. And that's it. I conclude. We found a correlate of consciousness awareness directly based on the concept of neural synchronization, which is a physical concept. And we see that the tendencies that state that has more conscious they tend to have maximal entropy or information. How do we plan to continue on this? Perhaps it's like looking at more subtle changes in consciousness, because we were looking at very rough changes like epilepsy, sleep, are very huge changes. So like it would be nice to see it in like cognitive experiments. And finally, I would like to thank the collaborators. Thank you.